STAT3-5 in cutaneous T-cell lymphoma. The study was done in Graz under the leadership of Professor Wolf, and the presentation will be given by Dr. Dai. Please.
Thank you for your kind introduction. So thank you to ESGR for giving me the opportunity to present our work in this stage. So I work with T-cell lymphoma, particularly cutaneous T-cell lymphoma, the leukemic variants of the cutaneous T-cell lymphoma. Just a second. Yeah, you can see the laser pointer bit. Okay. So I like to give you the study synopsis of our work. And here, the leukemic CTCL patients comes to our clinic. We collect the skin biopsies and also the blood samples from where we isolate the PBMCs from there. Okay, and try to, yeah, it's better here. So we try to collect the skin biopsies and the blood, and from there we isolate the PBMCs. From the PBMCs, we look at the TCR-V beta immunospectrotyping to find out the TCR-V beta positive malignant clones. Upon finding the malignant clone, we do cell sorting to enrich the malignant clone, and this enriched malignant clone, from that clone we isolate the DNA and went for shallow whole genome and whole exome sequencing. So initially we did the whole exome sequencing to find the recurrent mutational pattern in the leukemic CTCL patients, but unfortunately we were unable to detect any recurrent pattern, but we did find recurrent pattern from the whole genome sequencing data, where we see the common chromosomal aberrations in the patients, and mostly that STAT3-5 region gain, and along with the loss of STAT1 and SOX1 region. And these cells which we isolated, we use as in ex vivo for a drug screening to find that if this mutational these aberration status, if that is targetable or druggable. And also we established a mouse model in NSG mice and see that if our drugs or lead compounds really works. And also we checked in the ex vivo form, and we found that STAT3-5 copy number gains, the more the copy number gain, it correlates negatively with their IC50 value, which gives us a very nice therapeutic window. So moving on. So the STAT3-5 copy number gain are frequent in leukemic CTCL patients, and it co-occurs with STAT1 and SOX1 loss. So here I show a copy number analysis plot of one of our patients, and here you can clearly see that STAT5A, B, and STAT3 is upregulated, along with a complete loss of T53 and also loss of STAT1 and SOX1 region. And here is the log2 ratio, and here you can clearly see basically that there is around 74% of the patients indeed have STAT3-5 gain, 41% have SOX1 loss and also STAT1 loss, and 78% have TP53 loss, and around 56% of the patients have MYC gain. So with this genetic status, we further explored that if this elevated STAT3-5 copy number and the expression levels correlate with increased clonal proliferation, and indeed it does. So the more the copy number log2 ratio, the more there is blood clonality in the patients in case of STAT3, STAT5A, and STAT5B. And here is a clear picture of the healthy controls versus patients in the from qPCR, and in mRNA level, there is a clear difference in the STAT3, STAT5AB levels compared to the healthy controls. And here is a representative plot from two patients, patient 4 and patient 5, compared to the patient 6 who doesn't have a gain. And here you can see that these patients in the protein level also indeed express high amount of STAT3 and STAT5. Moving on, the malignant T cells infiltrating the skin. So we look at the skin biopsies, and here is also we see the same picture. Here you can see that phosphostat5 positive cells, and here is the CD3 positive cells, and here you can see that the CD3 positive cells and phosphostat5 positive cells overlay quite nicely in this dual staining. And here, indeed, the patients have a high CD3 infiltration, which is STAT3 positive, STAT5A positive, STAT5B positive, and the cells are also phosphostat5 positive. So this comes to the first part of the conclusion of my talk, which is that copy number gain of STAT3-5, which frequently co-occurs with STAT1 and SOX1, contributes to increased clonal expansion in leukemic CTCL. Then increased STAT3 and STAT5A, B expression is significantly correlated with the increased blood clonality in leukemic CTCL. Now, malignant T cells which are infiltrating the skin in biopsies. Also in the L-CTCL are also STAT3 positive. Now this comes to the first part. Indeed, we can use STAT3 as a marker for this disease, and then we check if this is targetable or druggable. And for that, we need some lines because the primary cells are limited. So we try to see that indeed the cell lines like 7, 8, myelin are usable like here. You can see that indeed these cell lines have a STAT3 gain and also STAT1 loss and so on. So we decided to use cell lines further for drug screening, and here is a picture. We use pimocyte, which was a USP1 inhibitor, but also STAT3 path, the classical STAT3 inhibitors, and some newly synthesized drug compounds from our medicinal chemistry collaborator from University of Toronto, and IQDMA. And here you can see that these JAN750, JPX750 and IQDMA have quite a low IC50, so we use them as lead compound for further investigation. And here you can see that indeed these drugs blocking phosphostat3 and phosphostat5 level, both of them, but we saw IQDMA quite having, despite of being a kinase inhibitor, it's working quite slowly. So we were a little bit suspicious of that drug, and that's why we checked still further that if these drugs, IQDMA and JANPIC750, if they work like how they work in cell lines along with HACAT and juvenile fibroblasts, because if we able to use these drugs ever topically, we need to check that if there is a therapeutic window and if they are affecting any toxicity on these cells, and indeed these drugs affecting quite nicely the CTCL cells, but not the keratinocytes or fibroblasts here in the gray or black bar. Moving on, as I just now saying that the IQDMA was having a slow effect on the cell lines, so we decided to do a kinome screen. And in the kinome screen, we did indeed found that IQDMA is actually hitting multiple tyrosine kinases like ABL, ALK, TYK2, JAK3, and also serine kinases like BRAF, PAK2, and PIM3. So we use the FDA available drugs of these particular targets and see that if they can mimic this IQDMA like phenotype, or like have IC50 like IQDMA. And indeed, we saw that not all other drugs, because they have a very high IC50, only the PAK inhibitor have a quite comparable IC50 like IQDMA. And here, so from there, we come to an idea, because PAK works as a nuclear translocator or nuclear shuttler. It catches the STAT3 dimer and shuttles into nucleus. So maybe blocking PAK is the main function, the main methodology how the IQDMA works. And that's why you use the IQDMA and do a nuclear fractionation assay, where you can see that upon usage of IQDMA, there is not change of phosphostat5 or STAT5 level in cytoplasm, but indeed there is no STAT5 or phosphostat5 in nucleus. So indeed, the IQDMA working through the PAK inhibition method. So here we compared the JANPIC750, which is a new STAT3 dual degrader, with IQDMA and a particular PAK inhibitor, and you can see that they are working in a comparable fashion, like they are inhibiting downstream MYC, PIM1, MCL1, cyclin D3, the downstream targets of STAT3. And then we checked if these three lead compounds also have an ex vivo effect on the patient-extracted cells, and here you can see that the higher the STAT3 copy number gain, the better the negative correlation, or the lower the IC50, and here that's applicable for STAT3 for both FRAX, the PAK inhibition, IQDMA, which is also working through PAK, and not so well for direct STAT3 degradation. Then we established our mouse model, where we took the NSG mice and injected the CTCL cell lines in the mice and measured their tumor growth, and then checked that how the drugs work on them. And here you can see that indeed these drugs works quite nicely in reducing the tumor growth. And here you can see that both the three drugs here bottom PAK, because it's shown differently because we have to use a separate vehicle for this drug. And here in the top for JANPIC750 and IQDMA, it's reducing the tumor perimeter. It's also reducing the Ki67 staining, so the cell proliferation, and also we did a CD31 vessel marker, and it's also reducing the vessel growth. With that, I like to come the current understanding from our study that in the healthy condition, the STAT3/5 pathway is normal, so there is normal level of STAT3/5. The STAT1 is there, which is actually competing with STAT3/5 to get phosphorylated, so the STAT3/5 also not hyperactivated, and SOX1 is also there, and it's not blocking the phosphorylation of JAK. So there is no normal cell growth, but in particular case of leukemic CTCL, first of all, there is a gain of STAT3/5. Then there is a loss of STAT1. So as a result, the STAT1 is not competing with STAT3/5 to get phosphorylated. And there is also loss of SOX1. So SOX1 is not blocking the phosphorylation of JAK. So there is a STAT3/5 pathway hyperactivation, which leads to the clonal T cell expansion. So what our drug is doing, so these small molecule inhibitors, either direct STAT3/5 inhibitor or particular PAK inhibitor or PAK kinase inhibitor is blocking PAK, or directly stopping the STAT3/5 by degrading the STAT3/5, or if it's blocked PAK, PAK doesn't able to shuttle STAT3/5 from cytoplasm to nucleus, which results in cell death, and this is shown in our mouse model, and also it shows in the ex vivo condition where it shows that quite high negative correlation. With that, I come to the conclusion that our findings demonstrate that increased STAT3/5 signaling resulting from the copy number gains on chromosome 17q and loss of STAT1 and SOX1 is a key vulnerability from leukemic CTCL, particularly Sézary syndrome, which can be blocked using drugs like Janpix 750, or it can also be blocked through PAK-mediated method in upstream kinase using drugs like IQDMA and FRAX597. So the clinical impact for our study is the same that what I was saying, like highly frequent STAT3/5 copy number gain in leukemic CTCL, which results from STAT3/5 overexpression and hyperactivation, might serve as a novel marker for the treatment and guidance and also disease monitoring from the leukemic CTCL. And further, the development of the characterized STAT3/5 degrader like Janpix 750 for further lead structure or the further lead structure improvement for IQDMA or PAK kinase inhibitor should be continued in research for the curative targeted drugs. With that, I come to the acknowledgement of my study, and I'd like to thanks Professor Wolf and our group, our human genetics department who did help us with the sequencing, our vet med, where there is another professor, STAT3/5 professor, and her PhD student help us, and all the places from where we collected the patient samples, University of Cologne, Würzburg, Innsbruck, Medical University of Vienna, where I stayed sometimes for the further research, University of Toronto for synthesizing the medicinal chemistry compounds, Centessa Pharmaceuticals also for their support, and the Janpix. Thank you for your attention. Happy to take questions.
Thank you very much for this comprehensive presentation. There is one question over there, please.
Hi, over here.
Yeah, hello.
Super nice talk. Thank you. The STAT3/5 degrader compounds that you have, are they degrading both activated and not activated STAT3 and 5?
Indeed, it's degrading the activated STAT3/5. So once the STAT3/5 got phosphorylated, it binds to the SH2 binding domain and it degrades. At the moment, the compounds were not commercially available. It's produced by our medicinal chemistry collaborators and the Centessa Pharmaceutical.
Thank you.
May I ask two questions on the copy number analysis? So one is that when we talk about gene amplification,
yeah,
we usually expect much higher copy number and much narrower lesion. And your data seem like it's more of a broad event with lower copy number. Like when looking at the log-to ratio, it was like around 2 or something. And when you usually think about colon cancer, we get like a mid-copy gene amplification, more than copy number 5 or something. So do you have any insights on the difference?
Yeah, so some of the patients which are like long-term, our mycosis fungoides patients, they are hypotriploidy, so they have a higher copy number, but rest of them, it's like there is focal amplification, so there is like around 2 to 3 copies, but not more than that, and particularly the long-term mycosis fungoides patients, they are indeed hypotriploid.
I see. And another question is, how did you determine the recurrency of the lesion? So, you know, when we dealing with copy number aberrations, we tend to do something like GISTIC analysis or something. And how did you determine these lesions are recurrent?
So here mostly my research on the leukemic CTCL. So the copy number changes which I showed is from the blood, not from any kind of lesional skin, but the long-term MF patients' lesional skin are also, we stain them, and there is the STAT3 gain. Is that answer your question, or did I miss something?
Yeah, thank you. Yeah, thank you.
Thank you very much.
Please.
Hi, Cristina von Almiral, and very nice talk. Thank you very much. So 2 questions. This PAX inhibitor, how selective it is, and if you have any knockdown assays of the STATs alongside.
Yeah, so the STAT3 inhibitors are quite highly selective. So I showed a graph that in comparison with juvenile fibroblast cells or Hackert, the keratinocyte cell lines, and indeed the STAT3 inhibitors are not hitting at all these fibroblast or keratinocytes, but it's very specifically hitting the CTCL cell lines. And further, when I show the data from my ex vivo data, where those particular patients' cells who have higher STAT3 copy number gain, their IC50 values are lower. So which gives us a more precision medicine window.
Thank you.
One more last short question.
Very short question, very interesting, thank you. So
Thank you
I have a question about USP1, which you were showing as an inhibitor. And do you have any evidence about the functional interaction between USP1 as a deubiquitinase and JAK-STAT pathway?
There is no I am aware that USP1 and JAK-STAT, but indeed the pimozide, which is a neuroleptic drug, it hits USP1. And in term also, it's shown that the pimozide have effect on STAT3 levels. So there are some publications from Niels Odam's lab in Copenhagen where they showed that they used pimozide and it reduced significantly the STAT5 phosphorylation.
But no direct interaction. For example, USP1 deubiquitinates STAT3.
Might be, I'm not aware of.
Okay, thank you.
Thank you very much. Thank you.
Thank you very much.
So our next